We are Group 6B, and our project will discuss RNA targeting small molecule drugs. Biological targets of drugs and hormones are generally enzymes, ion channels, and receptors. Coupled with the invasive processes of chemotherapy, radiation, and surgery, the inefficacy of protein-targeting drugs have prompted researchers to search for a new target. Small molecule drugs that target RNA rather than the enzymes or ion channels provide an alternative method to combat the pathogenesis of various cancers. In this presentation, we will explore the roles and structure of RNA in gene processing and regulation specifically related to cancer. We will then delve into the specifics of drug targeting, explaining how RNA targeting small molecule drugs offer a more efficient method of combating cancer and other diseases.
Before discussing the specific nature of RNA targeting molecules, we will discuss a few key points regarding the structure of nucleic acids. The chemical structure of nucleic acid is a heterocyclic base, a pentose sugar, and a phosphate group. The heterocyclic bases are nitrogenous bases, meaning they have amine and amide groups. A nucleic acid with two rings in its structure is called a purine, while a nucleic acid with a single ring is called a pyrimidine. The pentose sugar is made up of a core of 5 carbons numbered 1' to 5'. The phosphate group is attached to the 5' carbon. A sigma bond around the 1' carbon of sugar allows for rotation in the structure of nucleic acids. This means they can be found in two different conformations, either as an A anomer or a B anomer. DNA and RNA always have nucleic acids in the B conformation. Oligonucleotides are a series of dinucleotides. A dimer of DNA or RNA can be covalently linked with a phosphodiester bond between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of another. This gives a chain of nucleotides directionality from the 5' to the 3' ends of the sequence. Two chains of DNA can be held together by hydrogen bonds. The nucleotides that are able to bind together must possess the same number of potential hydrogen bonds. Because of this, the nucleotide adenine always pairs with thymine, and guanine always pairs with cytosine. The two hydrogen-bonded DNA chains form a double helix. The two DNA strands are complementary and antiparallel. Nonpolar, hydrophobic base pairings are in the center of the helix, while polar, hydrophilic sugars and phosphates are found on the outside of the helix. There are a few differences between the structures of DNA and RNA. While DNA's structure has one hydroxyl group on its pentose sugar, RNA's sugar has two hydroxyl groups. RNA replaces the nucleotide thymine with the nucleotide uracil, which is also capable of hydrogen bonding to adenine.
Nucleic acids are defined through their three structures. The primary structure is the linear sequence of nucleotides. It is essentially the order of the bases in the previously mentioned oligonucleotide. The secondary structure is determined by interactions between bases. In DNA, it is the hydrogen bonds between two strands. However, in RNA, a single chain folds back on itself to form pseudoknots and stem loops. The base pairs, like in DNA, form hydrogen bonds with each other on the same strand, leaving unpaired bases at the end. The tertiary structure is the large-scale folding of an entire chain into a specific three-dimensional shape. In DNA, the tertiary structure is a simple double helix, but RNA can have many different structures. This includes A-minor triplexes, where bases are inserted into the minor groove of an RNA duplex. In the case of adenosine, the base forms hydrogen bonds with one or both of the two hydroxyls of the duplex. Quadruplexes are formed when 4 consecutive guanines form a Hoogsteen ring by means of Hoogsteen hydrogen bonds. Since the hydrophobic surface of RNA is buried within the ring, the structure plays an important role in the binding potential of RNA by ligands. Positive metal ions, including magnesium and potassium, assist in overall RNA folding because they balance the highly negative charges of the phosphate groups. Transcription is the process of creating a strand of mRNA based on a sequence of DNA. The DNA strand that is transcribed is called the antisense strand, while the one that is not is called the sense strand. Translation is the process of using the mRNA sequence to create a specific sequence of amino acids that are used to create polypeptide chains and eventually proteins. Errors can occur through all of these processes and are the main source of genetic diseases like cancer. To understand the efficacy of drugs that target RNA, it is important to comprehend the relationship between RNA and cancer. Cancer is a genetic disease, the result of dysregulation of the gene networks that maintain normal cell growth. Even though there are both environmental and genetic factors to cancer, the majority of cancers are caused by somatic mutations that occur after conception. These DNA changes can include point mutations, changes in a long stretch of DNA, or epigenetic changes, the addition or removal of chemical markers. These changes can influence whether and how much RNA is produced. Even though only about 1% of the genome is protein coding, at least 20% show biological function and over 80% exhibit biochemical indices of function. These non-protein coding regions of the genome can serve as a template for the transcription of many non-coding RNAs. In fact, 90% of transcriptional output in humans are non-coding RNAs. There are both small non-coding RNAs and long non-coding RNAs. However, most RNAs affiliated with cancer are long, between 200 base pairs to about 100 kilobases. Some general effects of non-coding RNAs on abnormal gene regulation are microRNAs, oligonucleotide repeats, and post-transcriptional changes, or in other words, epigenetic modifications. MicroRNAs can induce gene silencing by binding to target sites on the target mRNA, thereby silencing genes that would normally inhibit cell division. Oligonucleotide repeats essentially are just the overexpression of oligonucleotides that contain a mutation, resulting in a disease state. Epigenetic changes can alter RNA structure and function without changing the sequence. For example, methylation occurs when methyl groups attach to DNA molecules, switching off the gene, thereby inhibiting functions that could regulate cell growth. A type of non-coding RNA specifically associated with cancer is incRNA. IncRNAs contribute to oncogenesis through the direct binding and isolation of various proteins. They do this through various mechanisms like silencing mRNAs that code for tumor suppressors, acting as complexes involved in RNA processing, thereby regulating the creation of mRNA itself, and acting as decoys of other mRNA molecules, attaching to a biomolecule it is not supposed to and preventing it from fulfilling its function.
Now we are going to explore the structure of drugs and how they are traditionally utilized to combat disease. Lipinski's rule of 5 is a rule of thumb to determine if the properties of a chemical compound make it a likely orally active drug in humans. The first component is that the molecular mass has to be less than 500 Dalton. This is because as molecular size increases, a larger cavity must be formed in water to solubilize the compound. The partition coefficient, or log P, which is the ratio of concentration of a compound in a mixture, has to be less than 5, since increasing log P decreases aqueous solubility, meaning there will be less absorption. There should be less than 5 hydrogen bond donors because the hydrogen bonds increase solubility, and they must be broken to allow the molecule to permeate through the lipid bilayer of the cell. Finally, there should be less than 10 hydrogen bond acceptors. Drug targeting can be generally grouped into two methods, passive and active targeting. Passive targeting consists of a nanoparticle that is coated with a hydrophilic substance such as polyethylene glycol. This allows water molecules to bind to the particle via hydrogen bonding, making it resistant to macrophages and able to stay in the bloodstream long enough to circulate. Active targeting is more specific. Ligands are attached to the molecule that recognize and bind to receptors only on the target cells. Both of the previous methods described rely on targeting some form of protein in an effort to mitigate the disease. However, there are a few issues with this. Many proteins are considered undruggable because they lack small clefts in which small molecules combine with high specificity and affinity. Moreover, the extent that proteins have been targeted is limited since less than 1.5% of the human genome codes for proteins, and only 0.05% have been able to be targeted. These limitations led scientists to exploring a different area, specifically RNA, which could potentially explain the proportion of genes that can be targeted. The fact that RNA is being considered as a potential target for drugs is a result of the evolving way in which we view RNA. It used to only be considered as the mediator between DNA and the protein it encodes for. However, now RNA has been shown to affect gene expression, and as previously mentioned, is even associated with disease. Moreover, the structure of RNA was thought to be indiscrete, but over the past 30 years, it was shown that RNA has definite secondary and tertiary structures that make it a viable target for drugs. As mentioned before, metal ions are used by RNA to counteract its anionic quality to help in RNA folding. RNA cannot fulfill its enzymatic responsibilities all by itself, so it utilizes ions to assist in its catalytic activity. RNA creates electrostatic pockets through its negatively charged phosphate groups to hold these positively charged ions, like magnesium. The ions shift the pockets to positions that activate the extra hydroxyl group on the RNA sugar by allowing it to release its H+ ion, making it more acidic. This allows the RNA to perform its function. Since these pockets of ions are located around the RNA's active site, the location of the catalytic activity, these pockets can be used as a target for small molecule drugs. What has recently been established as a way to target RNA is antisense oligonucleotides. These are synthetic polymers that are complementary to the mRNA that we want to target. This process works by identifying the sequence of disease-associated RNA and synthesizing an antisense oligonucleotide that targets that specific sequence and stops transcription. However, even this method has issues, mostly due to the large size of the oligonucleotides and their vulnerability to degradation by proteases, as well as the fact that this process is inherently not drug-like, making it difficult for them to be pharmaceutical agents. Small molecules may present a better way to target RNA because they can be designed to be more drug-like and have a better chance of reaching their target, mostly by oral administration. Also, small molecules can recognize the RNA by structure instead of sequence, which is important in non-coding RNA where the structure is conserved as opposed to the sequence to maintain function.
Yale researchers have found a method of targeting RNA with small molecule drugs. The researchers were primarily focused on identifying ligation sites using the tertiary structure of RNA rather than the secondary, which is the current method. This would provide higher specificity in molecular recognition of the small molecule drugs. They have focused their attention on pathogenic yeasts, specifically S. cerevisiae, which have become medically problematic for patients with compromised immune systems, like cancer patients. The researchers specifically set out to find a small molecule drug that would ligand to group 2 introns, large self-splicing ribozymes that are found in the mitochondrial genomes of plants, fungi, and yeasts. The researchers specifically sought to identify an area on the active site of the RNA, the area that actually catalyzes reactions, that could be used for viable drug attachment and was solvent accessible, which means hydrophilic. Metal ions proved to be the answer. As mentioned before, metal ions stabilize the tertiary structure of RNA, allowing for folding, thus hiding the hydrophobic areas of RNA within the fold. Furthermore, RNA creates pockets for these metal ions around the active site, perfect for drug ligation. The group 2 introns specifically contain a heteronuclear 4-metal ion center within the active site, so the researchers focused on the area around this catalytic center to find the area for attachment. Using an oligonucleotide substrate, the researchers first examined the RNA splicing action of the group 2 introns. Then they curated an array of 10,000 compounds to find compounds that inhibited the splicing activity of the introns. They then studied the effect of the functional groups of the selected compounds on intron inhibition in vitro, which means in a separate solution. After determining the most potent and inhibitory compounds, the researchers tested the compounds in vivo, in a live specimen, to determine its toxicity and viability. They found that the growth of S. cerevisiae in the presence of inhibitory small molecules is inhibited in a glycerol/ethanol medium, but not in a purely glucose medium. More importantly, they found that an intronless strand is much more resistant to the compounds, signifying that the intron inhibiting effect of the compounds plays an important role in stopping cell growth. The researchers ultimately found the highest potency in compounds 18 and 19. They named the compounds intronistat A and intronistat B, respectively. Testing these drugs on human cells, they found that there was no toxicity and no effect in general after a 24-hour incubation. This means that the compounds are highly specific to group 2 introns in particular. Even though this means that these drugs cannot be used for humans, it shows that compounds like these can be found and will show high specificity to targeted human RNA structures. Through this presentation, we have found that RNA plays an important role beyond traditional protein coding. Non-coding RNA plays an important role in gene regulation that propagates the development of cancers through changes in transcription and causing malformed cell division by various processes. We have also considered a more efficient method of drug delivery, targeted drugs, and more specifically the role of small molecule drugs in pharmacology. The coupling of research in non-coding RNA in cancer and the active targeting processes of small molecule drugs enable a more efficient method in counteracting cancer. As a precursor to cancer, RNA provides a burgeoning scene to delve into more research for cancer medication. Even though antisense technology that targets RNA sequencing exists, research into small molecule drugs that target the structure of RNA would provide a more efficient, easier method to, in colloquial terms, cure cancer. The research into RNA targeting small molecule drugs that have worked in yeasts offer compelling incentive to further this pharmacological method into reality for human cancer. Through this advancement, we can see that the curing of cancer is that much closer to humankind. We hope that through this presentation you have learned about the possibilities and chemistry of RNA targeting small molecule drugs. Thank you.